Okay, we're going to start out with chapter 3. We're going to talk about culturing microbes in the laboratory, primarily because to identify microbes, to study microbes, we have to be able to grow them in a laboratory setting so that we can work with them. And we'll talk about all the ways that normally this would be where we would start our live lab if we were doing a hybrid or a live laboratory class. We would start with what are the tools that we need? How do we work with microbes in the laboratory? So this is a little more sterile doing this online. You'll do some of these techniques in your virtual labs, but honestly, it's very different when you do it in person, and that's how you really develop the skills. So we'll do the best that we can here. So the 5 I's of culturing or working with microbes are inoculation, incubation, isolation, inspection, and identification. So we'll talk about all 5 of those. So inoculation is when we collect a sample and we put it on media, an artificial nutrient source. So we've inoculated the media and we have started a culture of some kind. Incubation is the time that we spend growing those microbes, usually at warmer temperatures. Sometimes we need artificial heat sources, incubators, to do that. Isolation, when we take a larger culture that may be mixed, it might have multiple kinds of microbes present in it, but we want to isolate down to individual genetic types. So we will talk about ways that we isolate cultures to get to individual genotypes. Inspection, we look at these microbes under a microscope. We use microscopes to help in identification of microbes and to look at qualities of microbes. And ultimately, in a microbiology class, your ultimate goal at the end of class is to be able to take a large group of microbes, have one sample that has many characteristics to it, and you use those characteristics to identify the microbe, because that would be what it would be like in a real lab, right? So you have a patient that comes in, they have some kind of infection, you need to culture that sample, grow it, inspect it, and then eventually identify it to utilize antibiotics, if it's bacteria, in an effective way to help control that infection. So producing a culture. So this is where we're going to propagate microbes, and we can use lots of different kinds of media or nutrient sources to do that. And then we're going to grow them. After we initially inoculate, we're going to grow them up. So what is media? So we'll talk about all these things interchangeably. You can have liquid medias that are in a tube. You can have media that's in a Petri plate. It's that usually substance that's like Jello that has a color to it that we can inoculate microbes on the surface of. Inoculation is introducing microbes to that culture. And then when we say sterile, we mean free from all life forms. No spores, no viruses, no other microbes contaminating that culture. We can usually assess sterile media by allowing a negative control to grow, where we don't open that media, we keep it closed, we don't inoculate it, and we can continue to assess that there's no growth on it, and that lets us know that it is a sterile culture. So incubation usually happens at temperatures of 20 to 40 degrees Celsius. We put them in an incubator where we can set those temperatures. A lot of microbes do just fine at room temperature, which can fit within that range. Some incubators, if we're growing human cell lines, and we'll talk about this, or animal cell lines, you need a CO2 injector into your incubator. And so if we're trying to culture viruses, the virus can't make what it needs. It needs cells to make those proteins to make more virus. So we have to culture viruses in a cell line. And if we're working with animal viruses or human viruses, then we have to do that in an animal or a human cell line, which means we have to do that in a CO2 incubator. Microbial growth in liquid media. We'll look at pictures in a minute, I promise. It looks cloudy. Sometimes there's a sediment in the bottom. Sometimes there's a scum or a layer across the top or on the sides of the tube. There's also sometimes a color associated with that. Microbial growth on solid media looks like piled up cells, visible masses that are growing usually on the surface of the medium. I do not want you to memorize incubation conditions for different bacteria. It would be something that you'd look up if you needed to know. But Listeria likes to grow at cooler temperatures. It is a common contaminant in dairy producing facilities, in food facilities. Listeria is a real problem with contaminated food production. So a lot of things get recalled for Listeria contamination. Pseudomonas fluorescens is something we found in the soil quite a bit, so it can grow at cooler temperatures. Things like Streptococcus pyogenes likes it to be a warmer temperature because it grows on or in humans. Okay, so this is what media plates look like, right? This is a Petri plate. You can kind of see a color here. Media may have a color, it may not have a color. We'll talk about that in a minute. But it's got this jello-like substance inside that we can culture microbes on. This would be a solid media. So we have to give microbes everything they need to grow in a medium. That usually means we've got a carbohydrate or a sugar source. It usually means we have a protein source. It means that we might have, usually we need water, right, to get this all into solution to begin with and then pour it out into a media. We need a gelling agent, which is usually agar, that acts like Jello, like a gelling agent to make it hard. And then we usually, depending on the microbe, will need different vitamins and minerals and sometimes other cofactors as well. There are many microbes that only need a few very simple inorganic compounds to grow. There are other microbes that are incredibly difficult to grow, that their media has 20, 30 different components to make. Think of it as making a really complicated recipe versus a really easy, throw it in the microwave with some water recipe, right? So some microbes can survive on very little, others need a lot, and it needs to be very specific. There are some microbes that can't be grown on artificial media, and they have to be grown in cell culture or in host animals, viruses being one of those. Types of media. So we're going to talk about all these. Some of this is obvious, right? So we classify media based on its physical state. What does it look like? We base it on its chemical composition, what's in there. And then importantly, we classify media based on its function. So these don't go together in any certain order, but these are the four physical states you can have. You can have a liquid, you can have a semi-solid, you can have a solid that can be liquefied, and then a solid that cannot be liquefied. You can have a chemically defined media that's synthetic, that where we know every single chemical and the amount of that chemical that we put in the media. It's very specific, and we usually use it for growing very specific microbes. Then we can have complex media, where we're adding some very rich media components that aren't really defined, and we can usually grow a broad range of microbes on those complex media. In terms of function, and we're going to talk about all of these individually, you can have a general purpose media that we use to grow lots of different microbes. We can have an enriched media that may have something special in it, like blood, that will promote the growth of certain microbes. We can have a selective media where we're growing certain microbes. We can have a differential media where when we grow the microbes on the media, they make a different phenotype, a different response. We can have a reducing media Those are media that are limited in oxygen for growing anaerobic bacteria that don't require oxygen. You can make media for transporting specimens around, for shipping specimens, and we have assay and enumeration medias as well. So let's just take a look at those. So liquid media, pretty self-explanatory, right? So they're water-based solutions. They don't solidify at temperatures above freezing. Things like you'll hear us talk about nutrient broth. You'll hear us talk about urea broth. Certain kinds of infusions, certain kinds of milks, if they don't get solid, then they're a liquid. There's some semi-solid media that are thick, but they don't make a firm surface. Not quite jello, right? A thick kind of gooey, viscous kind of substance. I don't use a lot of semi-solid medias. Solid media gives you a firm surface that you can streak out cells, you can isolate cells on. Those are the kind of media that we have in Petri plates. We use those for bacteria and we use those for fungi. Again, viruses are usually grown in cell lines. Those cell lines can be grown in a monolayer in a Petri plate, or they can be grown in a big flask of cells that are suspended in liquid. Agar is the magic substance. It comes from an alga called Gelidium, or a seaweed. So, and we use it to solidify our media. It liquefies at 100 degrees, which is great, goes into solution in the autoclave because we have to autoclave all of our media to sterilize our media before we pour it, before we turn it into a food source, a nutrient source for the microbes. And so we let it cool, and then we pour it into Petri plates. It solidifies at 42 degrees Celsius, and then we have solid media for plates. Most media that's solid is usually a 1 to 5% agar. 1% is usually enough, 5% is a lot. So this just shows you in its different forms. So obviously a liquid media, we usually do this in a tube so we can cap it so it doesn't run everywhere. A semi-solid looks thick and viscous. Again, we usually do that in a tube so that it doesn't run everywhere. We can do solid media in tubes. We can make slant cultures this way. We can make stab cultures this way, but we also do, so this here they're showing you as a solid reversible to a liquid. So there's one assay that we do that I don't know that you'll do in your virtual labs, where we assay to see if a bacteria has a gelatinase, which is an enzyme that breaks down gelatin, right? And so the media that we use for that, the nutrient gel that we use for that, is kind of a semi-solid or a solid that's reversible to a liquid. It's solid when we inoculate it, and then if the bacteria have that enzyme activity, it liquefies over time. So defined media is a synthetic media. Again, this is a media whose exact chemical compositions are known. Think of a very, very difficult, intricate recipe, because that's really what it is. It will usually have organic, carbon-based, and inorganic compounds in it. And every recipe will be very, very different. And it has an exact formula, and we use them to grow very particular microbes. I have a recipe book for defined synthetic media that's about 500 pages long, and if we're being very particular and trying to grow one certain kind of bacteria, then we may have to do this kind of media development. In all honesty, in a microbiology lab, you're most often using a general rich media that'll grow lots of kinds of microbes. And that would be called a complex media. So it contains at least one component that's not chemically definable. Well, how does that happen, right? So with some common things that we use in complex media, we'll use a digest of yeast, which means we grow up a huge yeast culture, and we dry that out, and we use that culture as a food source. We have no idea what proteins the yeasts were making that day. We have no idea of the specific chemical composition of that yeast extract. Same thing, we can do a pancreatic digest where we take a pancreas from an animal. We don't, the media companies are. They'll take pancreas, they'll dry it out, they'll powder it up, and we can use it as a media component. But we have no idea on any given time what proteins were present, what minerals, what vitamins were present in that pancreas. We know only that it is rich in nutrients, rich in proteins, and sufficient to be a food source for many, many microbes. It may have, like I said, ground up cells, ground up tissues, ground up secretions, but nutritionally rich. So some examples, a blood agar, a serum agar, meat extracts like the pancreatic digest I mentioned, a milk media, a soybean digest, very rich in protein, a yeast extract. Any of those would be considered a complex media and are very common to work with. So nutrient broth usually has yeast and a pancreatic digest in it with agar and sugar, and that's almost enough for nutrient broth. And there's enough there to support the needs for nutritional needs for many, many microbes. General purpose media are media that can grow as many different types of microbes as possible. So that would, a complex media contains that mixture of ingredients like we just talked about that supports a wide variety of different microbial life. Things like nutrient agar and broth, soy agar, a brain heart infusion would be an example, but not one we use very often. In contrast to that, an enriched media would be a complex general purpose media. However, it may have a special substance in it, like blood, like serum, like special growth factors, and that promotes the growth of certain kinds of more specific bacteria. You may have growth factors present in those medias that have specific vitamins or specific amino acids, and that too may enrich a media to promote certain kinds of microbial growth. So these are an example of enriched media. So A here is a blood agar, and we'll talk more about blood agar in a minute because it is a selective and differential media as well. But you can see here, here's our microbial culture that we've streaked on the surface of that blood agar. And you can see this clearing zone around the bacteria. What those bacteria have done have lysed those red blood cells that are suspended in that blood agar. So we've promoted certain kinds of microbial growth there. This is a MacConkey agar, and again, we're supporting the growth with this particular medium of gram-negative bacteria. So selective and differential media. So we'll talk about each kind because they have really extensive applications in biotechnology laboratories and microbiology laboratories. Here we have, and I think we're going to talk about each one individually. Let me skip ahead for just a minute. But we've got better pictures here. So let's talk about selective media on the left. My best example, when we're working in a biotechnology laboratory, in order to know that we're using the bacteria that have been genetically modified, we've usually modified them to make a protein that helps us select them, all right? So we'd make a media with an antibiotic in it, like ampicillin, and that genetically modified bacteria has a gene included in it that makes it resistant to ampicillin. So that when we streak it out on this ampicillin containing plate, we select for only the bacteria that have that ampicillin resistance trait, that ampicillin resistance gene. That's a selective media. A differential media is a media that gives us different results. So blood agar is a good one, and we'll talk about all the ways that we can select with blood agar. But here you can see blood agar that's uninoculated. Here you can see bacteria growing on blood agar on the surface, but you see that those cells are still present. Over here you see a clearing, right? It's not as red. So these bacteria are actually lysing those red blood cells. So that's telling us something about the bacteria that are growing on this kind of plate, because we're getting a differential phenotype in response to the media that we're growing them on. So again, selective media, we just talked about it, like antibiotic resistant media, they are going to have something in there that inhibits the growth of many microbes but allows certain microbes to grow. So it's really important for initial isolation of specific kinds of microbes. Differential media are going to give us differential phenotypes. So they allow lots of microorganisms to grow, but you're going to get visible differences. So down here on this plate contains a product called XGAL, which certain bacteria, if they have the gene, will convert that substance to a blue color. So this bacteria has it. Here where they've E. coli doesn't. And they've given you Staph aureus doesn't, and then Enterobacteria do on this particular sample. So you'll see differences in colony size. Color is a really important one with differential media. It tells us that bacteria have a trait that other bacteria don't have, and we can tell those bacteria apart. We can identify those bacteria based on the color they produce on that differential media. Okay, so blood agar. So it's an enrichment media. When we say fastidious microbes, we mean particular microbes, not all microbes. So we can use it as it's a rich media, but it's also a differential media. Certain bacteria have hemolysins, enzymes that lyse, break open red blood cells to release the iron that's in hemoglobin. So you can have bacteria that have beta hemolysis, which completely lyse or break open the red blood cells. You can have alpha hemolysis, where you have an incomplete lysis of red blood cells, or you can have gamma hemolysis, where you have no lysis of the red blood cells. So this is how blood agar can be a differential agar. So, and this is what that looks like. So with the beta hemolysins, you can see the bacteria in white here growing on the surface of the media, but then see the clearing zone. You can see that darker area, what looks darker but not red, around the bacteria. That's where those bacteria have lysed those red blood cells. Over here with the alpha hemolysis, you don't see a complete clearing, a complete lysis of those cells, but you see a color difference that suggests a partial lysing of those red blood cells. And then over here with the gamma hemolysis, you have bacteria that aren't lysing those red blood cells at all. So what we've done there is, if we have an unidentified bacteria, we streak it out on a blood plate, we get a certain phenotype there that helps us narrow down the type of bacteria that it might be. Some other media that are selective and differential. So we use MacConkey agar to identify gram-negative bacteria. It will allow the growth of gram-negative bacteria, and then it also allows for a color change based on whether those bacteria are good lactose fermenters or not. Again, we're trying to identify the bacteria. The MacConkey agar helps us to do that by being selective and differential. Mannitol salt agar is another kind of media. It has a really high salt content that blocks the production of most bacteria, but allows things that can tolerate a high salt concentration. It also produces a color change. So over here on the right, you can see those red colonies. So bacteria that can metabolize mannitol, which is a kind of sugar, when they do that, there's a pH change. And in mannitol agar, that allows you to see a red color, which tells you those bacteria are not only growing on the high salt concentration, but they have an enzyme that can break down mannitol. Reducing media is a media we use that drives oxygen off of the media continually and promotes the growth of anaerobic bacteria. Carbohydrate fermentation media, so that same assay we did with mannitol, we can do that in a liquid media for all kinds of sugars, for lactose, for dextrose, for mannose, for sucrose, all kinds of sugars, and we can assay bacteria to see if they have those individual enzymes. A sucrase to break down sucrose. When that happens, the media goes from being red to being yellow because there's a pH change that happens when that enzymatic activity happens. Transport media is a media we use that specimens travel well in at a variety of temperatures. Assay media, when we add we can add an antimicrobial drug, we can add an antiseptic, a disinfectant, we can add cosmetics to a media, and we can see how how a broad range of microbes respond to those substances. So that's an assay media. In industrial and environmental microbiology, we are continually counting the numbers of microbes that are present in milk samples, in water samples, in soil samples, to assay whether they are usually safe for human consumption. So we use an enumeration media that allows us to count more rapidly approximate microbial populations in any given commercial or commercial, industrial, or environmental sample. So a colony, we are trying to isolate usually individual colonies. Why do we do that? So we'll take a sample from the environment. It'll be a mixed kind of sample, lots of different microbes present, right? Here it's very clear. You've got a red bacillus type bacteria and you've got a white coccus bacteria. Well, we want to isolate those into individual samples. So we might streak a plate, and we'll look at isolation here in a minute, to get to the point where we have an individual colony. So we're literally thinning out the sample to allow a single cell, because bacteria, to allow bacteria to grow into one single colony that's all genetically identical. Yeah, so when we take when you take that initial inoculation and you put it in media, you're gonna get a mixed culture, and we're always trying to get that to independent cultures. So to isolate, to be able to get to that isolated colony, you have to have a really small number of cells in a fairly large area so that you can streak it out to thin out that sample. You usually do that on agar, on a firm surface, in a Petri dish, and we're gonna use an inoculating loop to do that. I believe you will have the opportunity to try this in your virtual labs. So this is a streak plate method, very common method, something we do all the time in the laboratory, where we would take your mixed culture, we would sterilize an inoculating loop, stick it in the culture, and then we would streak it out on a plate. Then we would sterilize our loop one more time again, and we would touch this initial culture one time and stretch it out, streak it out. Then we'd sterilize our loop again, and we'd touch that thinner culture one time and streak it out in another quadrant. And that's what they're showing you here. The idea is is to get the overall culture to a thin enough point where we have isolated colonies. You can see you have isolated white colonies here and isolated red colonies. Usually in a mixed sample, there would be many, many, many kinds of microbes in a mixed sample. A pour plate method is easier to describe with pictures, so I'll do that in just a minute. But essentially, we're creating a cereal dilution and plating that out on solid Petri dishes. So this is another way to get isolated colonies. It's also another way to enumerate bacteria, which we'll do at a little later time. So here's how we do that. We take our initial culture, we grow it in a tube, right? And we'd take a small sample of this, like a milliliter, and we'd put it on a Petri plate. We'd streak it out, and we'd see what grew. But then with that initial tube, we'd take a mill out of there, add it to 9 mils of media, mix it up, and then plate out another mill here. So we've diluted it by a factor of 10. And then then we would take a mill of this solution, and again, in a mill, 9 mils of sterile media, mix it up, and we'd plate a mill of that on a plate of agar. So we've diluted that again by a factor of 10 until we get to the point that we have diluted that culture enough that we can pick out single colonies from that culture. Steps in a spread plate are pretty much what we talked about, where you pipette a small volume onto the plate, and then you're going to use a sterile spreading tool, or what we call a hockey stick, which you may or may not see in your virtual labs. It literally is a piece of glass. You can see it here on the right that looks kind of like a hockey stick that we can sterilize in an open flame. We let it cool, and then we can spread that liquid culture around. Sometimes that's enough to get colonies that we can isolate. So a pure culture, once we get that single colony, we can inoculate it into a medium, where a liquid medium, and we can, or sometimes a slant, sometimes a stab culture, where it is the only known species in that culture. That's a pure culture. We use that frequently for laboratory study because we're trying to keep our cultures pure so that we know that what we're studying is the species that we're currently interested in. Anexic means that that culture is free of other living things except for the culture being studied. And sometimes we use subcultures where we'll use those pure cultures. We'll take a sample from it, and we'll take a second level culture from that well-isolated colony. Things that cause us a lot of trouble in the lab are mixed or contaminated cultures. So you can see here, they're showing you pictures of pure cultures, although, it's hard to tell from there. But you can tell the phenotype. They're all the same color. They don't look contaminated. When we have a mixed culture, it means somewhere along the way, somebody didn't sterilize their tools very well, and we've introduced two cultures, one culture into the other, and then we have a mixed culture that can result in a lot of problems once we start to work with them. Contaminated culture happens the same way. Contaminants can come from the environment outside, or we can introduce them. But when we plate these things, when we plate our pure culture out on a plate, and we see something like this white blob here that looks like it might be yeast contamination, and all of these red colonies, you can clearly see that something in the culture isn't right. So then we would try to isolate a colony and start over again to create a pure culture once again. So identification. So again, our final goal here after we've inoculated, incubated, isolated, after inspected with a microscope. So we do microscope analysis. So that's the inspection part of the 5 eyes. We are trying to get to identification as our endpoint, as our end game, right? In working with unknown microbes. We have lots of tools that we can use for that. We can use phenotypic testing, like we saw with the differential and selective medias, where we can look at phenotypes, color change, growth patterns, response to different chemicals that gives us a phenotypic test. We more and more and more and more, we can use genotypic testing. So we have a really handy way using a ribosomal RNA sequence that we can use a primer from that sequence and send off any culture for DNA sequencing. It's very inexpensive. We can do it for about $2 to $3 a sample now and get back enough of a sequence and throw it into GenBank and be able to tell what that sample is. That's why a lot, for at least for bacteria, a lot of the tools that we use in a microbiology lab, and a lot of the ones you'll use in the virtual labs, are actually kind of outdated because genotypic testing really gives us a more sure answer, and it's fairly inexpensive to do these days. With viruses, we need to use immunologic testing where we're testing antibodies and reaction to antibodies based on the proteins that those viruses make. Or on Western blotting, which we may talk about later. Macroscopic analysis, what do these things look like on a plate? Microscopic analysis, what do these things look like under the microscope? All of these things give us results that help us identify what the microbe is that we've assayed in the first place. Biochemical tests are another way. I showed you the sugar tests, that when there's enzyme activity, you get a pH change, so you get a color change. And there's tons of biochemical tests. There are ones that give off products. So they give you a color change or a media change. There are biochemical tests that screen for the presence of enzymes, again, where you get a color change. And you're going to do some of these as you go, probably within the first 2 to 3 weeks in those virtual laboratories, so that you can look at some of the biochemical tests that we use in a laboratory. You might use something like a citrate assay. You might use something like a gelatinase assay with a nutrient auger. You might use something like a thioglycolate that's going to tell us where do those microbes like to grow. If they grow on top, they're an aerobic microbe. If they grow in the middle, they're probably a facultative microbe, meaning they can grow in or without the presence of oxygen. Like I said, we use genotypic testing all the time, DNA sequencing all the time. And we also use that immunologic testing for things like viruses. Okay, so this gives you a chance to go back and review some of those concepts before we move on to the next section of Chapter 3 on microscopy.